We have with us Dr. Cedric Gondro. Dr. Gondro is a professor of computational and quantitative genomics in the Department of Animal Science at Michigan State University in the USA. His research focuses on the development of computational methods, particularly artificial intelligence, for the optimization of biological problems and genomic prediction using high-throughput genomic data, as well as its application for livestock production. His applied work focuses on genetics of beef quality, with a particular interest in the genetics of marbling. Current research topics include the use of artificial intelligence methods for feature selection applied to genomic prediction, the use of climate variables and computer vision to increase accuracy of phenotypic prediction in livestock systems, supply chain data integration between farmers and consumers, application of portable sequencing devices for livestock production, and understanding epistasis in the context of linear models. So thank you for joining us, Dr. Gondro. I'll give the floor over to you now.
Thank you. As you mentioned, I have quite a mouthful of a title. It's a professor of computational genomics and quantitative genetics. I'm usually known as just a guy who likes steak and plays with computers. So that's kind of what we're going to do today to talk about. We're going to talk about a little new gizmo that hopefully a little new gizmo that has really caught my attention, which is these portable sequencing devices. Just a second, I can't see my screen now. Man, this is being clumsy today to the extreme. Is that working now?
Yeah, yes.
Okay. All right, all I see is a blank screen here on my hand. Right, all right, let's try to be less clumsy. So I want to talk about breed composition with portable sequencing devices. It's really not so much about the application itself, it's more about the technology. And later on, we'll talk a little bit about some other things that I do on a daily basis. It's artificial intelligence for genomic predictions and a little bit of recent work we've been doing in enviromics. These are two kind of flavors of the month things, so it's always good to include these in talks as well. So as I mentioned, I'm quite fond of steaks. It's been something that's been a bit of a personal passion and a professional passion as well. We have been working mostly with high marbled premium beef. These attract high premiums at the industry level, of course. Things like Wagyus, Akaushis, Hanu. And just as a little bit of an aside here, at home I use these infrared grills. They've been great for barbecues, but we've also played around with these for quality taste panels, and they really do a great job of ensuring that there is homogeneity in the samples, and they do a good job of cooking things to the standards for tasting these in trained panel settings as well. Before we move on to all these lovely beefs, we just want to call your attention to this picture over here of the Wagyus. Keep this photo in mind, and we'll get back to that later on. This is the sample that we used to do the breed composition with our portable sequencing device. But before we get into that, just some background in terms of beef and marbling. Marbling is one of those important traits for meat quality. Depending on the paper you read, you will see that it's more or less associated with other quality trends, but it is a big driver of quality in beef. It's associated with tenderness, juiciness, and flavor as well. Again, depending on the literature you will read, these associations are not so obvious, but in some samples we worked with, these correlations were very high. These are just phenotypic correlations in samples of about 500 animals. So it's a reasonably small data set. It's direct phenotypic correlations. But we have over here the juiciness, tenderness, flavor going in the right direction, which is you want them to have a positive correlation with marbling, which is your intramuscular fat over here, and has a negative correlation with shear force, which is hardness of these steaks. And we see that marbling is really quite a strong driver for palatability ranking in consumers as well. This is a smaller data set. I will say that this really is a reliable data set. It was more of a little internal test exercise, but we gave tasters 4 different random samples from different marbling scores. Each tester got 3 out of 4, and the marbling scores was 9+, 7, and 8. So 9+ would be our highly marbled kind of extreme Korean, Japanese breeds. 7, 8, also these high marbled animals, but a bit less. Then 5, 6, and then 3, 4. And what we do see is that consumers really rated the palatability in terms of 1, 2, and 3 based on the marbling. So the higher the marbling, the more they prefer this kind of steak. So marbling is one of the big drivers of palatability, and it is something that I personally like myself as well a lot, personally and to work. But there are no real decent quantitative measures for beef quality. There's a lot of noise in the system. Some countries are a little bit better, other countries a little bit worse, but I don't think there's a single country in the world that really has a very decent metric which is objective in terms of beef quality. So we get bombarded with a lot of sources of information. So breeds, Angus, Wagyu, Wakaushi, etc., traits, grass-fed, grain-fed, natural, healthy, dry-aged, wet-aged, if they are organic, ethical concerns, welfare. So there's a lot of noise in the system. There's also those grading systems, choice, select, prime, A5, Kobe beef, etc. So none of these really provide sufficient clarity for decision making. It has a lot of great obfuscation value, but it's not really ideal for accurate decision making. So we were trying to build systems, and one of my main interests is building systems that will allow us to get something a bit more quantitative on these. So depending on your personal values, you'll be able to make more informed decisions on these. Nowadays, products, by and large, they have more stickers than a backpacker's bag. And we're trying to get something that's a little bit more informative and not just this generic, mostly marketing side of things. Things, for example, like Kobe beef. Our local butcher over here, one of our local butchers, they have up in the cabinet something that says Kobe beef. It's probably just an F2, F3, or a small percentage of Holstein with Wagyu. It's not a Kobe beef as per definitions of Kobe beef. And of course, the price is matching it. It's quite a cheap product as well. But on the other hand, you see this label, Kobe beef, at a low price, and you see Kobe beef at about 20 times the same price in a different place. And it's really hard to make any decisions based on this kind of information. But so where we started was to look into things like breed composition and product certification. When we started thinking of applications for these portable sequencing devices, I got fascinated by the technology. It's such a little tiny gizmo. It's portable, it's cheap, and it seemed very interesting. We said, oh, this kind of a product could be used in a range of scenarios out in the field. So we don't not depend anymore on laboratories. We don't need to send samples to labs. We can kind of take possession of genomics for our own rights. And that's kind of where it all started. And we thought that a nice, simple way to start with this would be with breed composition and product certification. So basically, it's just grab a sample and do the sequencing and really see if it is what you kind of purchased or what you think it is. Breed composition is not a particularly challenging problem, especially if it's just sorting out if this is 100% or if it's not. It can be more challenging if there's multi-breeds involved to get the exact percentages, but if it's just an exclusion test, it's not a complicated problem. We've been doing this for quite a while since the SNP chips came out. And it has quite a fair bit of applications, of course, for branded products, to make sure that they are what they are, for breed integrity, certification, authenticity, provenance, especially when you're talking about expensive premium products. If there are allegations about a product, they can be tested and see if they really are the real product or if they are counterfeit products. Just a bit of an aside here. This is from another project, but it gives an idea of what goes through processors. This was samples collected in one day on an abattoir for some other work we were doing. But we did the genotyping and we had a look at what kind of breed composition of the animals we had going through the abattoir that they had. And as you can see, there's a lot of variation between Angus, Holstein, and Hereford, which is mostly what we have over here. And they are crosses. So we have some that are 100% Holstein, others that are mostly Angus, some background in Herefords. So there's a lot of variations in what is going through the abattoir. These will be ranked into choice, select, prime, and they will be side by side in shelves at the local butchers. There's really no distinction between these later on. But that's just a bit of the background. This brings us to the Minion. This is a nanopore technology, and this is the sequencer. It's just this tiny thing here. It's a handheld sequencer, very portable, reasonably easy to use, costs quite little. It's just about $1,000, so it's quite cheap as well. And the main advantage of this for us when we were thinking of playing around with this was that we would not need to send samples to the lab for genotyping anymore. So it's kind of taking the genotyping process straight into your own hands, where you could do this in the field itself. And we wanted to see how practical that was and if it would work or not. Okay. In terms of technology, it's a little bit different because it produces long and ultra long reads and not just short reads like the Illumina or other technologies. So that's what we did. We tried this out at my place at home. We had a home sequencing party. We set up a little portable kit set, so some portable gears for spinning machines and the heaters and coolers. We did the DNA extraction right on my kitchen top at home. This was the beef that we extracted, so of course we were forced to eat all the rest of it, but did not go to waste, of course. And so the whole steps can be done by untrained personnel. None of us have any training in molecular biology. We've not spent time in the lab and managed to do the DNA extraction, the library preparation. And then we run the sequencer on a desktop on top of my coffee table at home. The whole process takes about 6 hours. Takes a bit of time, but we went from getting the sample all the way to the DNA extraction, library prep, sequencing, and breed composition prediction in about 5-6 hours, 6 hours more realistically. So what about that sample? It was an F1, so it was not a straight Wagyu. It's a cross between Wagyu and Angus. The company confirmed, and that's mostly what is sold as Wagyu in the US. And there's little differentiation between F1s and pure Wagyus. I would prefer, of course, a bit more clarity on these aspects, but that seems to be the general industry standard right now. In terms of applications and limitations of these, so when we started out, it's basically you can do with this whatever you could do with genotypes. So if you can send it to a lab for genotyping, you can do the same things with these technologies. Main difference is, of course, now you don't need anymore a lab or specialized personnel, but it still does take a fair bit of practice. So there will be trial and errors and wasting money in the process until you get the knack of it. So it is not simply plug and play. Especially the DNA extraction, if you have no experience in this field, it can be a bit trickier. Applications, of course, is whatever you can do with genotype. So breed composition, testing for recessives, estimating breeding values, etc. Theoretically, this should take around 4 hours. I would say realistically, it's more like 6 hours to do it. The applications in the supply chain, again, where we were thinking of in terms of a regulation certification of breeds, provenance, etc. Food safety issues for DNA signatures. Of course, you will need a database with these signatures of the original animals, but it could be based for tracking back sources of problems. Now, it does have some downsides. As I mentioned, it does take a bit of practice, but it's not extreme, and we figured this out on our own, just reading the manuals without. We wanted to take a more amateurish approach to it and see if this could really be taken to the field. The reagents are not room stable, and they have short shelf lives. So it's not just something you can chuck in the shed and forget until you need it. It will require planning to be able to use it. The DNA extraction is always a bit trickier than we would like. It's not sufficiently automated to make life simple enough. It is right now more expensive than genotyping. And the data structures need to be in place for the analysis, of course. If there's no data structures, then it's just a bunch of ATCs and Gs in a FASTQ file, which have rather little value. So we have to develop online databases that can be, where the data can be uploaded to be useful. Now, I think what was the greatest downside of this was that it's great for a few samples, but it does not really scale up. So we're talking about 6 hours per sample. Okay, if you have a small farm, 5, 10, 20 animals, you might be able to get through that. But if there are thousands and thousands of animals on a farm, it really does not scale up easily. To require batteries of minions and a whole lot of computers to be able to do this. So it didn't seem to be really to have the portability we had hoped for in this, or the scalability we would have liked to see. But what I'm still very convinced that it will work well is for disease testing. We're trying to develop some applications in this space now. And ideally, you'll see a scenario where vets or farmers who have this in their pocket. And if there is any risk of an outbreak or any, let's say any questions about what is going on the farm, they would be able to just do a nose swab or a pooled nose swab, extract samples from blood or even fecal samples, run these through the machine and get a diagnostic in a reasonably short period of time for diseases. So we're trying to develop these more on the computational side, the disease database, to be able to test these, and methods to be able to quickly do the analysis for this. So this is an application which I still think is quite convincing. Another group that's working with this is Ben Hayes in Australia. They have put out quite a bit of work in this space. If you look at their work and their papers, they will have a very different feeling to it than what we do. They are much more, let's say, enthusiastic for on-site sequencing applications. They have taken a very professional approach to it. So it's more molecular-based, developing protocols, and we have tried to steer away from that being the complete amateur. So what we do doesn't always work. It's much more expensive. So there are these two trade-offs, but what we wanted was something that would be easily applied in the field and not something that's another method that You can take the method, but you still need to take the technical stuff and the technical knowledge along. So these are the two sides of the arguments on the nanopore sequencer for field applications. Now, we did some other animals as well, some wagyus, akaushis, and also the commercial beef sample that we saw. We also genotyped these samples and sequenced with the flogo for some comparisons. In terms of working with these data sets, it's pretty much the same as any sequence data. Call those bases, convert to FASTQ, then map the sequences to the reference, call the variants, do some quality filtering, and then you can do the breed composition. So it's reasonably straightforward, but there are a few quirks in the process as well that we'll discuss in a second. And some of these, first for us, we got a lot of variation in the amount of raw base reads that we got. We went from 1.3 million to 202 million reads. So a lot of variation what you get. Again, always considering that we are amateurs in this space. We're not molecular biologists. We're just computer people playing around with molecular techniques. And also the depth of reads is trickier here. We don't have enriched spaces where we're amplifying certain regions. These are all random sequencing at very low coverage. And since it's low coverage, there's very low repeatability of what gets sequenced. So if we're filtering out at a depth of just one single read count, we had a variation between 32,000, which is very low SNPs, and these SNPs coming from alignments to the reference data set, up to about 5 million. So we go from less than a 50K SNP chip to 5 million. At depth of 2, we go from very low 180 SNPs to 550K. And at DP3, we go from absolutely no calls to just 50,000 calls. So DP3 is more or less the kind of bare minimum we would like to have to be able to do some quality filtering on these reads. But if you get to this depth, you will have nothing left in the data. And also you get rather sparse genomic coverage because these are longer reads. So some chromosomes are well represented and some are completely not represented at all. This, of course, posed a bit of a challenge for read composition, because if we simply do our variant calling based on the depth of the reads, if we call the reads with at depth 1, the variant callers will call these as homozygotes. They're going to be either the reference or the alternates. And this, of course, is wrong because we have only one of these nucleotides. So that basically makes the data collapse to the center. So we cannot really work with the genotypes in this scenario. We have to work with the haplotypes. And that's still work that we're doing to improve on this. It's I'd say probably gimmicky and maybe more technically interesting rather than applied interest, at least right now until these technologies become cheaper or faster. But we've been looking at computational aspects for the read composition using haplotype data and also to speed up these processes. So the raw data to variant calling is actually quite fast. So just about 4 minutes on the GPU, so it's pretty straightforward. But if you want to then use these variants and subset genotypes of reference data sets, it takes a bit of time, especially if you're going to try to read BAM files directly. These are pretty slow to work. So we're thinking of writing some tailored code for that. And right now what we're doing is going from Plink to VCF to WAR, kind of off-the-shelf analysis, but we're thinking of moving all of this into an HDF5 structure, see if that would speed up a subsetting of the reference to align the haplotypes for the read composition work. And these, of course, are a little bit on the big side, so you end up with some rather large matrices of about 5 million SNPs by 40,000 haplotypes. That would be a reference on which you want to do the read composition analysis on. So we're working a bit on the computational aspects of improving this. And also, we want to move a bit away from this model so that it becomes independent of the actual SNP set that we use in the data. and independent also of reference haplotypes. So something that is more of a probabilistic-based approach, given the data and conditioned on the probability of the haplotypes in reference data sets. And try to get point estimates plus some uncertainty around these breed composition estimates. So that's kind of where we are at right now with this portable sequencing devices. Okay. And just for comparison, we also did a full genome sequencing of an individual, this chap over here. We ran 7 full arrays on these. This is the kind of coverage that we were getting per array. So we can see that we've been improving. So it is a learning process. We started with a very low coverage in the first one, and the last one was pretty okay. We got a reasonable depth over these 7, but at the end of the day, this ended up being quite an expensive exercise. It would have been much cheaper just to send it out to a core facility to do the sequencing. We spent about $7,000 on this year, and the sequencing itself for even higher depth would be about 2,000. So moving on a bit, some other things we've been playing around with. Well, maybe not just us, but a lot of people have been kind of following this little recipe here, which is we've done a lot of sequencing of key individuals. We have a lot of historical genotype data, and people have been essentially imputing from these lower density panels up to the sequence level, and then doing work on genomic selection, with GBLUP, single step, BASE-R, association studies, and so on at the sequence level with imputed data. It's computationally quite intensive, of course, because we increased the number of SNPs from 50K, 100K to 10 million, 8 million, sometimes even more, depending if it's a multi-breed population, you end up with about 30, 40 million SNPs. So it does become computationally quite intensive. And over here with the little pig, it's just a little plugin for our own work. If you need to do any imputation on pig data, we have a swine imputation server hosted here at Michigan State that will take in the 50K data and up impute it to sequence level. And that's free to use, of course. So what's the good side of doing this, of imputing up? Oh, here, same data, 50K, 700K, and sequence data. What we do see is that we get better resolution. We do not really improve our confidence around the QTL. So the uncertainty around where the QTL is still more or less the same, but it does give us more clarity on where these QTL peaks are. But to me, at least, what I most like is that it helps better separate the foreground from the background. With 50K, we don't have much separability. We see where there's a background, but there's a lot of noise also over here in this region. Once we increase to sequence level, we start to get a bit more clarity on regions that might be interesting, even if we do not have enough power to estimate them accurately. For example, this region over here, it's not significant, but it's definitely a region that might be worth having a look at. Now, the bad of this is that basically it doesn't really make too much of a difference for genomic prediction if it's the business as usual models. So we already knew from theory that increasing from 50K to 700K to sequence would not make much of a difference to our prediction accuracies. So that's what theory said, and we saw the same in real populations. So by and large, imputing up has not really improved our prediction accuracy. There are papers out there. It does improve a little bit with multi-breed systems or for predicting breed A or breed B, providing these two breeds are reasonably close to each other, but still independent. So it does improve a little bit on that side. But by and large, it doesn't really help much. And that's mostly because our mixed model equations are essentially just conditioned on our GRM, on our genomic relationship matrix. And after 100K, the changes to the GRM are rather minimal. as you can see here, between 700K and sequence data, the only differences are really across the diagonal. And it is these marginal differences that will create any differences in the EBVs, because we're putting in a slightly modified GRM over here, which will give us slightly different EBVs. So the prediction accuracies don't improve much. It's just a 1.5 from 50K to 700K, and then 0.6 to sequence. This was in a very closely related population. So you might wonder, what's the point? It's just a big computational exercise. Oh, the point is that in a perfect world, we would actually know the true SNPs that are associated with a trait and the true causal variance. Yes, that's probably rather realistic still, but it's something we can aim for. And what sequence does, it's, at least in theory, most of the functional variance, or most of the variance, should be inside the data. So we do now have a chance of actually identifying these. So in that sense, there is some opportunities to explore with imputation. And if we also note these large variants and all the small variants, we will use only them for prediction. For example, in this example, which is a very Mendelian trait, we can see this is with imputed sequence data, and we see here a very big signal. So if we use just these two top SNPs, we will do a better job at prediction than using the entire data set. Of course, this is the magical GWAS that we all would like to see, but we hardly ever do. Again, it's a Mendelian trait, it's horns in sheep, but that would be the power of knowing the truth, when we can use the truth instead of trying to average these out across population structures. So we can think in terms of a trait G, where instead of using the full GRM, we are using only the functional variance to create a trait relationship matrix. And then we use a normal GBLUP approach for that. And in simulation studies, that does a pretty good job of improving those prediction accuracies. Now, how to identify these is, of course, a bit the question. What's the best method to identify these functional variants and remove the noise? So we've been thinking in terms of feature selection approaches for this kind of work. This is really separating the noise from the signal. Now, the signals are very conditioned on the data itself and what kind of genetic architecture we have in the trait. If the traits are less polygenic, we should have larger signals, and we have a better chance of identifying the true variants and selecting those for breeding purposes. Now, if there's a lot of genes involved in the trait, the effects are small, then of course it's going to be very polygenic, and the background and foreground are going to be very confounded with each other, which will make it hard to pick up signals and subset the correct panel of functional variants from the rest of all the other millions of variants that are non-informative on the trait. And just to illustrate this point, I know this is a rather ugly graph, I'm afraid, but we looked at different genotypes. So we used real genotypes here: pig, cattle, sheep, human, and random. Random is completely unlinked data, and different numbers of QTLs, 10, 100, 1,000, and 10,000 QTLs mixed in with the other genotypes. And what we see is that if we start using just the truth, we will do a very good job of predicting. But as we started adding in, based on estimated SNP effects, the other SNPs, we start losing prediction accuracy over time. And this will eventually stabilize at what is our current using all variants for prediction. So when we kind of flatline in these lines is when we are hitting our current GBLUP approaches. And on the top is if we knew the truth or started adding these. So what we wanted to explore is how can we identify these top SNPs? And also note that we don't only start at the top. When we have a lot of QTLs, we will not be able to estimate these effects, even if we know the truth, because we just don't have enough power to get these effects correctly estimated. So this will depend then on the population size, the relatedness, and number of QTLs in the sample as well. So it's not just about identifying them, but also being able to get correct effects, given that you know the truth. So One of the methods we tried out, which works reasonably well and kind of reflects that previous signal-to-noise ratio plots, is this iterative weighted GBLUP approach with local search. I think we can call this the kitchen sink approach to genomic prediction because we just throw everything at the problem. But essentially, it's about splitting the population to three parts: a training population where you train the model, an internal validation or internal testing which you use to optimize the model, and then, of course, an external data set to see that things are actually working well. The principle behind this is to do a weighted GBLUP, and then we iterate these weights until they converge. So the difference between the normal G and the weighted one is, over here we're optimizing these weights based on the prediction of the effects there. So we scale the effects that are estimated in different rounds until this converges. So this is just your standard weighted GBLUP. Then once we have a first pass, we get the rough number of SNPs, and we try to see how these are working on the accuracies of the sorted SNPs. And then we go and do a local search on this window that we think is the best interval, so SNPs around here, and we try to kind of brute force approach, putting in and taking out. If you take it out, it reduces the accuracy, you put it back in. If you take it out and nothing happens, you leave it out. So we try to find the optimal subset. And when we're done, we refit the final model with this subset and evaluate on the external data set. So this is a way to reduce the computational burden of using full sequence data, where we can just go down again to smaller SNP panels that are hopefully just as accurate or more accurate than using all the sequence data. How does this work out? Again, simulated data with different population structures and genetic architectures. It kind of reflects what you see with the signal-to-noise ratios. The more discrete these traits are, the easier the signals can be picked out, and the better we do with our job. So when we have just 10 QTLs, we always do a very good job out of it. When we have more QTLs, we start going worse. So as soon as we have, let's say, 10,000 QTLs in the model, then we are really losing our ability to find the signals and separate them from the noise in the structure. And then your typical GBLUP approaches work much better. Okay. We tried many different approaches for machine learning as well, for genomic prediction, your multi-layer perceptron, convolutional neural networks, differential evolution, extreme gradient boosting. And by and large, our results matched up what everybody else is coming up with. That sometimes it's a little bit better, sometimes it's a little bit worse, but it never has really been groundbreakingly better. We had only one example where we were really happy with it, which was with differential evolution, where we tried, let's say, complicating more this differential evolution. And this is real data. We started with the GWAS, then we did a BASR, then a GBLUP, and this is what our prediction accuracies nowadays in the industry are more or less at. And then we tried some of these different differential evolution-based approaches. And we got really good results with this, close to the heritability estimates. So we're basically at our theoretical maximum of prediction ability. So this got us really enthusiastic about it. The problem was that this method worked on this particular data, but it did not really extrapolate into other data. So with other data, again, we were back to this, sometimes a little bit better, sometimes a little bit worse. Now, what people have been always talking about is that these machine learning methods will be great when there is epistasis. Right now, most papers finish saying that it would be great with epistasis, but they have already used real data in some part of the paper. And by and large, the results are sometimes better, sometimes worse. So if there was epistasis in these real data sets, these models should, in principle, have picked them up already, which they really haven't. So that doesn't seem to be the most convincing argument. But we tried to create completely epistatic models. And we went to a very extreme example where at 6K. This is not pairwise, this is a 6-way interaction. So it's very complex interaction models where everything is genetically determined. There's zero environmental noise, but there's no additivity. And the GBLUC predictions are very low. Our heritability estimates are only 0.09, and our prediction accuracy is 0.10. And then we tried different epistatic models as well, your normal G by G, G by G by G, and so on. They also did not really improve the prediction. But the deep learning was not a magic bullet either. We did much better, but this was really fudging the data. So we went out not to try to test the method, but we said, okay, let's try to do whatever we need to get better. And so it's kind of a cheaty approach that was used, and we managed to push this up to only 0.25. So we're still at just a quarter of the total genetic variation that's in the data, and we're still not capturing this with these models. But possibly they could do a bit of a better job. And just to wrap this up, so just a little nod into genomics, enviromics, integratomics, and some other dataomics. We're also looking a little bit into THI and changes in THI over time. We're working with the Angus Association on this project, and we're looking at what is happening in terms of THI. THI is your temperature humidity index. It's a measure of a temperature resilience in animals, or how of getting affected or not. And we're not looking just at the THI itself, we're looking at changes in THI over time and the number of outlier events in regions. And what we have observed is that the number of outlier events are increasing quite dramatically over the decades. So when we go back to 81, 90, there was not too many outliers, but now in 11, 20, this has really increased a lot. So there's a lot of THI increases over time. And this is not THI across the country. This is THI at the location. So it's an increase in extreme THI events in that particular location. When we look up here, we see oops, sorry. Let me come over here. When we look at these events, we see a concentration of extreme THI outlier events over here in this region and some over here as well. This does not mean that the northern US has the same number of THI events as Florida, of course. It just means that the number of events in Florida is increasing, as are the number of events over here, where you have more high THI events than you had in average in the past. And these regions seem a bit more stable. How does this affect production? So it actually does have a bit of an effect on the phenotypic trends. If we look at the differences, it is affecting production. So animals who have more of these and more exposed to these high THI events have lower production outcomes. So the calves are born earlier, the weaning eggs are different, and there seems to be a tendency towards increasing these as time goes by as well. So it does have an effect, and a quantifiable effect on the outcome of individuals. So what we're thinking is that we can use this kind of information also for phenotypic prediction in association with the genotypes. And of course, eventually we'll be looking into G by E as well. Other things we've been looking at is hyperspectral imaging. We're looking at using hyperspectral data for meat quality prediction. So we're trying to set up a little project where we'll be doing marbling scores, meat composition, fatty acid, IMF, and doing some imaging phenotypes and then associating these as well. Ultimately, we want to have a completely integrated system from consumer to farmer, where we can do predictions with these cameras instead of having to do the lab predictions, but also we can connect farmers with consumers to get two-way communication. So the farmer can feed in information to the consumer about what they're doing with the operations, what breeds they have, what kind of quality there is in the meat, to inform consumers, and consumers can also give feedback through this chain on what their perception of product is and what their values are. We've developed a blockchain for this, Again, this is probably more academic than in practice. Putting these things out and getting them adopted are sometimes more political than technical, but the technical work behind this we have completed recently. And one of my students did a very nice job of integrating this supply chain. He's working now on trying to measure methane emissions that are accrued through the supply chain system from farm all the way to consumer. That's a bit of a proof of concept of these applications. And I think I will leave it at that. I've already gone over time, mostly due to my own inability to operate the technology. This is just a summary of what we are currently working on and our main interests in the group. And of course, there's always a lot of people involved in these projects, and I basically just get to talk about things and do a bit of computation from time to time. And I'll open it up for discussions, questions, and comments. Thank you.
Thank you, Dr. Gondro. If anyone has any questions, feel free to raise your hand, or if you would like, you can turn on your, or just enter the question into the chat as well. I can start. I was wondering, for the nanopore technology, is there any limitations to data storage, or how would you go about, yeah, for the data storage if it was for like personal use on a farm or something like that?
It's not too big because it's low pass. So it give me 1 second, I'll just check on our computer, give you the exact file size, but it's not the worst. We're just looking at about 7 gigs or so.
Okay.
It's not the best.
Is that for an individual?
Yes, the raw data.
Okay.
But our group here, this year we went over 1 peta in storage space. And that's just here. It's once we moved into sequence data, things became rather ridiculous. It's the best word for it, frankly.
Yeah. And for the for AI genomic prediction, do you know kind of the reason why it's not quite as accurate as the GBLUP methods or the BLUP methods?
Oh, it's not better, it's not worse. It's sometimes a little bit better, sometimes a little bit worse. So
right.
I say it's comparable. And the short version is BLUP is really, really good.
Yeah.
It's a solid it's a solid method. It works well.
Right. So what would be the benefit of switching to a AI algorithm for prediction, if there is any?
Right now, there's little convincing arguments for it. We will have to find maybe better ways to manage the sequence data in the same way as moving from 50K to sequence for a GBLUP is largely useless. It's moving from GBLUP to AI is going to be largely useless unless we find better ways to do it. I think we can still make some more progress in this space and maybe do a little bit better. But things like feature selection can play a role in this area because we're moving from 8 million SNPs, and instead of, if we can identify the main variants, that even if they're not doing particularly better, but they are at least reliably the same, we can subset it down to smaller sets. And these potentially can hold also across generations, so they might hold a bit longer before the prediction accuracies decay. So we might have less erosion over time, and also might work better for less related individuals. This is something that we're still trying to test out and see how far we can push these things, but that's kind of where we are at right now.
Okay, that's interesting. Would that, because I guess the kind of a main problem with the single step or the GBLUP is the inversion of the the H matrix. Would that kind of help with that kind of?
It solves that problem, but creates other ones.
Okay.
So with the AI, once you have a prediction model, it's better. So the prediction models are quite fast and they're portable. So that would be okay. It's like having the snip effects. Once you have the snip effects, just multiply by the genotypes and it's easy enough if you want to go down that path. So think of it in terms of snip effects prediction instead of having to create those big matrices. Now, while we don't have to worry about all those inversions, all this runs on GPUs. And you need a lot of computational GPU grunt to be able to train these models.
Right. Right. Okay. That's interesting. Thank you. I think we have time for one more question if anyone has one. Flavia, go ahead.
Hey, Cedric.
Hey, Flavio, how are you?
Good, good. How about you?
Good, man. I don't see myself in this stuff.
Good presentation. Thanks for doing this, Cedric.
I was hoping to go over to you, but didn't get a chance.
I have a question. So the results that you presented from your group comparing the different methods, that was for the purebred animals, right?
Purebred animals, yeah.
Yeah. So if you had to analyze crossbreds, would you change your expectations about the behavior of like AI models or?
I haven't tried on different AI on different breeds, but in one of those examples, the sheep populations were multi-breed populations. So they're very close connected to each other, but they are separate populations. Although we just did randomize those, so I did not try to kind of separate and predict A from B.
I
would not change too much the accuracies of prediction, but I haven't really tried that out.
Yeah. No, I was more curious when you have really crossbreds in your system to try and also so when you identify features that are consistent across the breeds, right?
Yeah. Oh,
and if you could really identify them, then the prediction will be improved. Right? That will have a higher impact than in a single purebred population, right?
Yeah. But even if it is a crossbred population, then it probably is okay. I don't think it will make any difference if you're trying to predict A on B so much. You know, let's say you're trying to use Angus to predict Herefords. Those distances are probably too great. Maybe the feature selection on the sequence level gets a bit closer to whatever it actually behaves as additive, but still to be shown.
Yeah, no, I was thinking that when you like to do selection with animals that are crossbred, you don't like to infer the purebreds, but no, within the crossbreds. Because if originally, for instance, the marks could have a different phase with the, no? So in that case, you're not going to predict well, but you could find these markers that have a consistent phase, right?
Yeah.
And then place more weight on them. You could have a better prediction.
Yeah, that's what we're hoping, that in a perfect world, if you have enough samples, you should be able to pick up at least that stuff that works. And with sequence data, you get close to variants that are functional or semi-functional, or very tightly linked, and those should port well across, pretty much across all breeds. Right.
Hopefully. Yeah, okay, thanks. I will give you a chance for others to ask questions. Good to see you.
Good to see you.
Okay, any more questions? Or I think we are at time. So thank you, everyone that joined us today. And thank you again, Dr. Gondro, for the really great presentation. It was very interesting.
Thank you. Thanks for inviting.
Yes.
Apologies for the startup mess.
No worries, no worries. Sometimes Teams we've had some problems now and then, so. It's all good. So thank you again, and thank you everyone. See you all next week and have a nice weekend.
So have a good day.